It gives me sort of great pleasure to introduce Susan, who I've been working with for the last year on Team Pagona. Susan, she did her master's in Berlin on the dynamics of vitamin B in the plant Arabidopsis. So that's the white rat of the plant world. And then she moved on to do her PhD in Prague on the regulation and translation initiation in yeast and human cell lines, which is hardcore stuff. And then extended that to a postdoc after she finished the PhD. And then, for whatever reason, decided to come to Australia, where she worked first at the ANU in the John Curtin School, where she sort of worked around translation regulation and other aspects of gene regulation, and finally came to UC to work on our model species, Pagona viticeps, looking at basically gene regulation in relation to sex determination, and in particular the influence of temperature and how it reprograms the regulation of sex determination. So I just finished by saying Susan's one of these rare individuals who's actually worked in the lab as a wet lab scientist, and also progressed into bioinformatics. And so she's a bioinformatician that has a very deep knowledge of genetics, and that's a really good combination, and she's been weaving some incredible magic with Team Pagona. So I'm really looking forward to hearing your talk, Susan. I'm sure everyone else is, so pass over to you.
Okay, thank you, Arthur. That was a very nice introduction. So the title of my talk, you can see it. Can I first ask, can everybody see my pointer, this red dot that I'm moving here?
Yes, we are.
Oh, thank you. Okay. So the title of my talk is The Integration of Transcript Abundance and Translation Efficiency for Gene Expression Studies. And so it doesn't sound much like ecology. So my talk is also not about what I did here at UC, but it's more about my previous things and how to integrate it into what I'm doing here, basically. So so I'm not an ecologist, as you heard from Arthur. I just joined the Institute for Applied Ecology a year and a half ago, so I really don't know very much about ecology. But I do know something about gene expression studies because this is what I did. And what I'm also still doing here, but in a different context. So as Arthur mentioned, so the X here basically kind of relates to the current topic. This is sex determination in an Australian lizard. And we are indeed using gene expression studies to learn something about it. And I found this wonderful graphic in a publication in 2015. It was a review, and it just shows that so gene expression studies, they are used at least in some areas of ecology. So it's not far off. And probably as in every other field, the usage of RNA-seq just increased massively, I would say, in the last years, because it became just much more available. Mainly because it also became much cheaper. So, so first of all, I would like to give you an introduction into gene expression. I found this nice little schematic on the internet, and I think it depicts all the steps quite well. So usually we think about two major steps if we think about gene expression, and this is the step of transcription and also the step of translation. So in this schematic, you have some more important steps mentioned, and that is quite fine. So, and if I can just walk through you very quickly, gene expression starts with the DNA. So one gene at a time, let's say, is transcribed into an RNA molecule. And this RNA molecule still contains exons and introns, but for the final gene, we only need the exons. So the introns, the green bits in here, they are spliced out. And the red bits, they are combined. And then we end up with what we call a mature messenger RNA molecule. This molecule also gets a poly A tag in the end, and it gets a cap in the beginning. And then it's really ready and mature. Then it will be exported out of the nucleus into the cytoplasm. And this is the place where it gets translated. So a peptide is made, and the peptide will be folded into and assembled into a protein. So, and the protein is actually the functional unit, if you want so. So the protein is the actor. The protein does the job. So, for example, just to relate to the current topic, sex determination, this protein could be aromatase, an enzyme that converts testosterone into estrogen, and therefore the amount of this enzyme and the activity of this enzyme is quite important for the decision if an embryo turns into a male or a female. Or this protein could be a transcription factor. That means it could go back into the nucleus and act at the level of the DNA and decide or basically be important for how much of a certain target gene is transcribed. Yeah. And so when we do gene expression studies, very often, at least, we would actually like to know how much of this functional protein we have in the cell. But what we use in order to estimate it is the mRNA level. So we do RNA sequencing. And we do it because it's just very convenient to do. It is easy to do, and it is relatively cheap to do. And we use this as basically an estimate for the protein level in the cell. So as you can imagine, there's a lot of regulation going on. Gene expression is a huge cascade of steps. There's a huge space for a lot of regulation. So there will be regulation going on upstream of RNA sequencing. For example, here I just listed some points that will influence transcription. It is by far not all, but let's say it's the most important points. So everything what happens upstream of RNA sequencing, so all the consequence of all the regulation happening upstream is accounted for in the RNA sequencing result, of course. But all the regulation that is happening downstream is, of course, not accounted for. So translation is one of them. So translation is a hub for a lot of regulation. So this will not be captured, of course, by an RNA sequencing experiment. But translation is not the most downstream step. There's regulation also going on downstream of translation. For example, there can be protein folding, assembly, protein localization, modification, degradation of protein. So there's really plentiful steps and regulators in gene expression. But anyway, for this talk, I would like to focus on translation. And I tried, this is just a very intuitive thing. The more downstream the regulation happens, the faster will be the response to the stimulus. So even on the level of, if any regulation happens on the level of translation, it's still a relatively fast response to a certain stimulus that a cell encounters, whereas regulation that happens on the level of DNA is usually a more adaptive kind of regulation. And so I searched the literature a little bit to find information about how well mRNA levels can predict or are correlated with protein abundance. And this is just something from a review in 2009. So it's a little bit old, but it just shows that it is not the same in every organism. So it does vary. So this, and there is a certain correlation. So it's not too scary. The correlation is not perfect, but the correlation is there. And in humans here, the correlation is not too well, but just a study done 2 years later. And this study was done really very proper. They showed a little bit better correlation between mRNA levels and protein abundance also in humans. And what the study also tried to assess is the predictive power that the different steps in gene expression have towards the protein abundance. And they see, for example, that the transcript level has a certain predictive power, but a little bit more predictive power has the translation, like the translation. What is not too surprising because it's simply closer to in the cascade, it's closer to the protein, to the final outcome. But nevertheless, so I think you can appreciate that the bulk of genes, or most of the genes, they have a very similar correlation between mRNA and protein, but there are still a lot of genes that have totally different correlations. So not every gene is just the same. I think this is more or less the message. That's why sometimes it is worth looking into more than just the transcript level, let's say. And another thing that is quite intuitive to understand why mRNA level and protein level are not always going together is that there will always be a time delay if there's a rearrangement going on in the transcriptome. This, for example, happens during development when a cell is set on a certain path. Usually the transcriptome or rearranges itself, like genes are switched off, other genes are switched on, and the protein levels, they will be delayed in this. But also if the cell has to correspond to a certain stimulus or to a change in environment very quickly, this is very often done on the level of translation. So the mRNA level will just stay the same, and it will not be visible on the mRNA level because just the translation efficiency of the mRNAs that are there will be increased and more protein will be produced, but not visible on the mRNA level. And so here I would just like to give you, I think it's a very nice example how translation, a very nice example for translational control in my current field, in the field of sex determination. This is an unpublished work, and it's not done by me, it's done by a collaborator in the University of New South Wales, by Paul Waters. And so what you see here is the X chromosome or one of the X chromosomes of platypus. And you have, so the X chromosome of platypus has two different regions. It has an X-specific region, and then it has a so-called pseudo-autosomal region. But let's focus on these X-specific regions, on the X-specific region. So you see that the transcript level, the average transcript level of the genes here in red is indeed lower in males than in females. But surprisingly, or I guess to the surprise of everybody, later on they looked at the protein levels and whoa, the protein levels, they were just the same. So there's no difference in the protein level between males and females. So what one would guess what's happening behind is that these lower amount of transcripts are just translated with higher efficiency. And this translation efficiency accounts for the lower level of transcripts. But the final outcome, the protein level, is the same. So, and this directly brings me into the world of translation, the wonderful world of translation. And this world wouldn't exist without ribosomes. And the structure of one ribosome is depicted here. Ribosomes are just huge particles made from proteins and RNA, and they have a large subunit. This is here depicted in gray, and they have a small subunit that is here depicted in purple. And they also have, what you also see here is the mRNA molecule here in green, and it's just threaded through the ribosome between the small and the large subunit. And these two guys here in the middle, these are tRNAs, but you can totally forget about them because we will not need them for the rest of the talk. Okay, so here I'm showing you, because I like these pictures, I'm showing you an electron microscopy picture from of a polysome, basically. What you see in the picture is a single mRNA molecule. It runs kind of in the middle here, and it is bound by I don't know, more than 10, 20 ribosomes at the same time. And all these ribosomes simultaneously translate this one mRNA molecule into proteins. So 10, 20 proteins are made at the same time. So you see the ribosomes as the black dots and the proteins coming out of the ribosomes. Yeah. And so just in a schematic, it would look like that. So this is the mature RNA with the cap and the poly A tail. And every RNA, every transcript has 3 parts, let's say. They have in the middle kind of a part that will be translated into the protein. So this is the coding sequence. This is the sequence that makes the protein basically. So this is the sequence where these elongating or translating ribosomes are sitting, and they all move in the same direction, and they all make one protein per ribosome. But then there's these 2 untranslated regions, UTRs, at the end and at the beginning. They are called the 5' UTR and the 3' UTR, and they have regulatory purposes, actually. So seeing this picture, I think you can very easily appreciate how it can happen that the same amount of mRNAs can very easily lead to a totally different amount of proteins in the end. Or how a totally different amount of mRNAs can also easily lead to exactly the same amount of proteins in the end. So, and this now brings me to a new method that I just would like to mention during this talk. Like, the method is not new, it was published around about 10 years ago, but it's quite a wonderful method, I would say. And so this method can tell us how many ribosomes are sitting on one mRNA molecule and where they are sitting even. And this method is, what's so wonderful about the method, it's that it's easy and relatively cheap. So, and I think this is a little bit the take-home message of this talk. There is an easy and relatively cheap way, it's similar to RNA-seq, to get this information, actually. So the difference between RNA-seq and ribosome profiling is actually only one step in the protocol, I would say. So RNA-seq takes these mRNA molecules, right? So usually in RNA-seq experiments, we use this poly A tail to purify out exactly these type of RNA molecules to get to separate it from all the other kind of RNA molecules in the cell. So we take those and then we make the library and we sequence it. So this is the workflow of an RNA-seq experiment. So for ribosome profiling, it's very similar. It's one step more. In the beginning, or traditionally, a drug would be used. It's called cyclohexamide. This drug just stops every translating ribosome in place at a time. So it provides a kind of a snapshot, a translational snapshot of the cell. But nowadays, this drug is not used anymore if it can be avoided. Like, it can be used, of course. It creates some biases that are known, so it's not a harm to use it. But if there's a possibility to snap freeze the cells or the tissue in liquid nitrogen, then this method would rather be chosen over using cyclohexamide. But nevertheless, so the one step that comes in addition to RNA-seq is actually an RNA's digestion of all the unprotected mRNA. So all the mRNA in between the ribosomes will be digested away by a huge RNase treatment. The ribosomes themselves, they are such a huge particles, they actually stay intact, they survive this kind of RNase treatment. Of course, it is a balanced kind of treatment, but we end up, so all the mRNA gets digested except for the little bit piece of mRNA that sits in between the 2 subunits of the ribosome and that is protected by the ribosome. And this we call a footprint. We make the library and we sequence it. So it's relatively simple. And so the result would look like that. For example, for the schematic drawn here, you would get, say, 3 footprints for the 3 ribosomes sitting on it. And very often, these results are depicted in a heat map. So then only the first nucleotide of this footprint is actually depicted in the, in the heat map. And these footprints are usually around 30 nucleotides in size. It differs slightly from organism to organism. And I hope you can see, so there is actually a 3 nucleotide pattern in this heat map. I'm not sure if you can see it. We call it 3 nucleotide periodicity. If you cannot see it in the heat map, maybe you can see it in the slightly different depiction down here. So this is because a ribosome that translates, it moves 3 nucleotides at a time. Yeah, it moves really codon by codon. And it's very nice that we have this resolution in the data in the end, because that means that the digestion of the RNA around the ribosome is actually so precise, or the generation of the footprint is so precise that we can get these 3 nucleotide resolution in the end. And what's good about it is that it gives us also some confidence, and that the ribosomes we are looking at are really actually translating the mRNA. They are not just sitting there for fun, or they just bind randomly, but they are actually moving in this pattern, so they are really translating. And so the very first study, so the first time this was actually introduced to the scientific community was in 2009, and I found in their supplements an estimate about how well in their experiment the mRNA density correlated with protein abundance, and how well ribosome density correlated with protein abundance. And it is no surprise, of course, that the ribosome density would correlate a bit better with protein abundance. But this is not the real, so the real benefit actually from Riboseq comes from combining the Riboseq with an RNA-seq experiment. Because then we can get all kinds of information about what kind of regulation is going on in the background. For example, if we have the same mRNA level, so the mRNA level doesn't change, but the amount of footprint has changed, then we know this is a gene, or at this time, only purely translational regulation was ongoing. For some reason, the translational efficiency was down-regulated or up-regulated. Or another example, if we have exactly the same amount of footprints, but the mRNA level was changing. That means basically that for some reason, the cell changed the translational efficiency and tried to account for it, or to compensate it by a different, by changing the mRNA level. So the mRNA level was adjusted to the change in translation efficiency, and the outcome is basically the same. You get the same amount of protein, and so on. So there's all these kind of regulations can be seen if we combine these 2 experiments, and we really see it. So if you plot all the genes, you can see that all kinds of possible combinations of regulations are going on. So these are already 2 good reasons, I think, for such a Riboseq experiment, let's say. So it can a bit better predict protein abundance, but combining it with the RNA-seq, we get this information about where the regulation and how the regulation is happening. But there are even more benefits from it. And I would like to present now 2 experiments, 2 examples from my current kind of work, or from our current kind of work in the group, where Riboseq can be would be really helpful actually. So one example are 2 genes. They are called, or proteins, they are called KDM6B and JARET2. These 2 proteins, they are so-called chromatin modifiers. So they act in the nucleus on the DNA. That means once they are made, these are some proteins that will travel back to the nucleus, and they will change the property of the DNA. So they make the DNA more or less accessible for transcription factors. So basically they decide if a gene opens, if a door is open for transcription, or if a door gets rather a bit more closed for transcription. And these two genes are quite important for embryonic gonad development. So the gonad, of course, plays a role in sex determination, so that's why we are interested in it. So these two genes, I guess they generally play a role in development, but especially also for gonad development. But further, they especially play a role in sex determination under temperature-induced sex determination. So under the influence of temperature, these two genes, they have a certain, let's say, decisive power in if an embryo turns into a male or a female. So they play a role in it. And so let's look at their transcripts. So let's look what's going on there. So the pre-mRNA is depicted here. So the pre-mRNA has exon and introns still in there. The introns are then spliced out. The whole molecule is kept and polyadenylated, and then we end up with a mature mRNA. If we see a transcript like that, we have no second thought. We think, of course, yes, this is translated. We don't think about it. But if we see unusual transcripts, then we don't know sometimes, will this be translated or not? So, and this, for example, is happening with these two genes. We get a second type of transcript. These transcripts, they retain an intron. And now there's the question, yeah, what's going on with this transcript? So transcripts that retain an intron, they can be kept at the nucleus, basically as a kind of reservoir. So they are there, they are waiting until they are needed. And if they are needed, the intron can just quickly be spliced out, and the transcript is really ready and mature, can be transported into the cytoplasm to the place where translation will be happening. But we have really good reason to believe that this transcript is actually really translated as it is. And but we don't know. We can only suggest or speculate at that moment. And if it was translated, then it would produce a protein that is just a little bit shorter than the normal protein. It's missing a little part here in the end. The reason is that this intron introduces what we call a premature stop codon. So the translating ribosomes, they would need to stop here. They can't go on. So, and it happens that this little piece of protein is actually a protein domain. It's just exactly one domain that will be missing, and it's a zinc finger. And one could think that having or not having this domain might give the protein a different target specificity, for example. So, yeah, but we don't know. Yeah, we can only speculate. But Riboseq could very easy, for example, answer this question. If we imagine these two transcripts are in a 50-50% ratio in the cell, and if we do Riboseq and we get a signal that looks like that, yeah, it's just the same, let's say, over the full range of this open reading frame, then we would say, okay, only this first transcript is transcribed. But if you get a signal that looks like that, for example, it drops in the end here by 50%, this would be a good, let's say, evidence that both of these transcripts are really translated. And there's a second example with exactly the two genes that's going on, because they produce a third variant, like a second strange variant of transcript. So this transcript, it seems it's much shorter than the normal transcript. And also this transcript, we have strong reason to believe it plays a role in actually temperature-induced sex reversal, because it just appears in an extremely high amount. And interestingly, even though the gene is shorter, still it would have what we call an open reading frame. So every protein starts with an AUG. And if there wasn't an AUG in the middle here somewhere, and this couldn't be translated at all by theory, then there would be no question. We would say, yeah, okay, it must have a different, this transcript must have a different reason. But no, there is actually an AUG here. So it's, again, kind of a strong hint that it is most likely translated. Or it is just a strange coincidence or whatever. But Riboseq could actually answer this question. So again, we can imagine we have these two transcripts in a 50-50% ratio. And if only the first one would be translated, again, we would get this even signal over the full length. Whereas if both are translated, you have a drop in the signal in the beginning. Yeah. So this is how, and Riboseq will, like RNA-seq, of course, it looks at all transcripts at the same time. So it gives information about the whole transcriptome. And in the end, I'm not sure how I'm doing with time because I wasn't monitoring it, but oh no, there is still time. Okay, sorry, I was irritated. So this is a third example from my current kind of field, right? From sex determination in Pogonaviticeps, the Australian lizard. So this is a gene that's, it's called NR5A1. It's a transcription factor. And this gene is quite important to us because for long it has been now a candidate as so-called master sex determining gene, let's say. So a master sex determining gene sits always very, very on the top of some cascade. It basically is able to switch either the male or the female pathway on. So, but we don't know really the master sex determining gene in Pogona, but we think this could be it. It is a transcription factor. So again, this protein will travel back to the nucleus and it will influence the transcription of their target gene. And what's going on with this gene, with NR5A1? So what we see there is we end up with two different kind of pre-mRNAs. So the one pre-mRNA has one intron and exon more in the beginning and the other one has one less. So this is actually not a very unusual situation. It happens quite often. Let's say so many genes, I would say, have this feature. I couldn't quantify many, but I have seen it often. And it's and we see it in Pogonaviticeps as well, like this. And what does it mean for the mature RNA molecule? It actually doesn't change the protein at all. So this open reading frame that contains the protein that will be translated, it's exactly the same. But what changes is the 5' UTR of the protein. So in the one transcript, the 5' UTR is shorter. In the other transcript, the 5' UTR is longer. And for somebody like me, having worked in translation, regulation of translation, this is a very interesting feature because most of the regulation of translation efficiency happens in the 5' UTR. So you can envision it maybe, so here I zoomed a little bit in. So why most of the regulation happens in the 5' UTR? It has to do with how translation is actually initiated. So I zoomed into the 5' UTR here a little bit just to be able to show you. During translation initiation, only the small ribosome subunit binds to the mature mRNA, and it binds really at the very 5' end. And then it has to travel all the way along the 5' UTR until it reaches basically the AUG, the start codon for translation. And when it finally reaches it, the large subunit will bind, and then the full ribosome basically can start translation. But a lot of things can happen while this 40S is actually traveling along here. And we know, for example, that when we need unregulated high translation efficiency, this 5-UTR will be very short. And this is, for example, the case with ribosomal proteins, because the cell needs a massive amount of ribosomal protein. And to have a short 5-UTR is just a safe game because there is less sequence where something can happen to the 40S while it is traveling to find the initiation codon, the AUG. So what could be happening is, for example, there could be structure. There could be a highly structured region and the 40S has really problem to go through it, or it can't and it has to stop. It might fall off if the binding between the 40S and the mRNA is weak. It can just not make it and fall off in between. What also can happen is that it skips an AUG entirely. So the design mostly is like that the first AUG that is seen, the small ribosomal subunit will stop and wait for the 60S to join, but it not always happens. So if the AUG is in a weak sequence surrounding, it can be skipped, yeah. And the last feature that is quite interesting is that there might be tiny little open reading frames sitting in the 5-UTR. So they don't make a useful or functional protein, and they very often have only 1, 2, 3 codons, just a few, but they can capture the small ribosomal subunit. So if the ribosomal subunit sees this AUG, it will initiate here. So it will here bind the 60S subunit. It will translate these 1, 2, 3 codons. It will stop here and then it will dissociate. So usually these UOFs are traps, and they will lead to a very low translation efficiency of the protein, of the real protein. But again, under certain conditions, it magically might happen that this AUG is not seen and the 40S will just scan on, and then it will increase the translation efficiency of the main protein. Anyway, wouldn't it be wonderful? So now we can see the footprints of the full ribosome. We can see everything that's going on within the part that is made into a protein. Wouldn't it be wonderful if we could actually get the footprints of these 40S small ribosomal subunits that are doing their job in the 5' UTR where all the regulation is going on? And so we thought, yes, it would be wonderful, and we tried to do it, and we did it. It's now published for yeast and also human cell lines, and it's called 40S ribosome profiling. It's actually not very different from the full ribosome profiling. Very often a crosslinker is used in order to freeze everything in place. So this is the most laborious part, because for every single cell line, for every tissue, for every organism, this crosslinking would need to be optimized in a very good way. The better optimization you get with the cross-linking, the better are the results in the end. And further down the protocol, there will also be a 40S and 80S separation be happening. They can be easily separated by their density. And that means it will give information about the 40S footprint separately from the ADS footprint. And these two data sets then can be integrated. So we see translation efficiency down here going up or down, and we can see what up here might have been caused this change in translation efficiency downstream, and so on. So it's quite lovely. And also, of course, we can add a co-immunoprecipitation step, because a huge amount of factors is involved in this kind of phase, the initiation phase. And when we pull down on a certain factor, we know when in time or when in place this factor is actually coming, going, and acting. And last but not least, I would just like to give you two examples of two results. This is maybe too much into the details, but it will be quick, and these are the last two slides. So in yeast, for example, one of the prime genes that is so heavily studied because it has an insane translation efficiency regulatory mechanism, and this gene is GCN4. So basically, this is again a transcription factor. It has a function in stress response, and because stress response always has to be fast, this mRNA of this transcription factor is always high. There's always a lot of mRNA around, but this mRNA is not translated. So there's no protein basically made. And this has to do with these upstream open reading frames that you see in the 5UTR here. Yeah, anyway, what we found, because we could for the first time look actually at the 40S trace over this, over this area. And what was interesting is that there is a and next to what we thought we would see, we also saw that there's a little structure here where the red bar is. And exactly before this structure, we see a lot of 40Ss just lining up. And they are kind of a reservoir, basically just being there, ready to go, but they can't go yet. But something under stress conditions will change, and they are just ready to go and translate this mRNA. So this was for our dudes in translation initiation, this was quite nice. And another thing, very similar, actually a gene with the same function as GCN4, just human. This is ATF4. Same thing, it's a transcription factor. It acts upon stress, and it has high mRNA levels usually in the cell, but no protein is made, and only as a stress response, there is some protein made. And also here we found, and this was surprising, that there is a little tiny kind of a U-off very at the 5' end of the transcripts, and they are kind of, they seem to be a trap or also a reservoir for these 40S ribosomes, for the small ribosomal subunits. We don't know exactly the mechanism of it. They definitely have translation factors bound, so they are ready to go, but the story is not entirely told in this case. So this is the last thing I wanted to share with you. As a summary, basically what the talk, what I hope I could transport to you in this talk is that protein and mRNA levels, they show a reasonable correlation, but integrating both types of data, if it is so easy as with combining an RNA-seq and a Riboseq experiment, can actually give us really exciting, insights basically into what is going on. And yes, it will work towards our understanding of the principle of gene expression control. So that's it for me. Thank you very much, everybody, for listening.
Oh, thank you very much, Susan, for a great talk and pitching it at a level that we can all understand, as opposed to being highly sort of using a lot of jargon. That was really good. I can understand it. So if I can understand that, I think a lot of people in the audience will be able to understand that. So thanks very much for that. Do we have any questions? Just switch mute off if you're asking, just in case. Well, maybe I'll start while you're thinking of one. So we're using northerns to get an assessment of the relative abundance of the isoforms. What you're suggesting, I think, should complement that quite nicely. In fact, I'm wondering whether we need to do the northerns if you've got this technique for looking at the sort of relative abundance in the context of the ribosomes for assessing the sort of relative abundance of the different isoforms that are functioning in the cell.
So are you asking for like us, for our group, or?
Yes.
But I mean, the thing is, you know, this Riboseq experiment is relatively simple and easy. And yes, my suggestion is, why not? We could just give it a go. Yes.
Yeah. Okay. And
it's basically.
It would complement the organs, right? It wouldn't replace them. Yep.
I think so. It would be a good complement. If you know, it needs, of course, some time and some money. It's additional money. So it's like doing 2 RNA-seq experiments at the same time. But so it will be twice the amount of money because you need twice the amount for sequencing. But from a technical difficulty kind of thing, I don't think it adds much technical difficulty, especially in our case, because the organs, as I understand it, they are snap frozen relatively quickly. And this is all you need for a Riboseq experiment.
All right. Any other questions? Let's see. Are there any PDFs?
Can I ask like a technical question?
Oh, of course.
You were saying that like the gonads just need to be, or I guess whatever tissue, but I always think about gonads.
Yeah, of course.
Do they, you know, the embryonic gonads are itsy bitsy, teeny weeny? What is because we're already kind of at the technical limits for RNA-seq. Is it the same sort of thing? Or is the low input a problem, and it would only be possible maybe on like adult tissues or juvenile tissues?
Yes, so I mean, you use now 1 gonad for RNA-seq, so there's nothing left of this gonad, I think, to do another experiment.
No, it's hard enough getting what we need out of
exactly.
one single gonad. You can pull both gonads together from a single individual.
Yeah.
And then a lot of people do pool multiple gonads across multiple individuals, but I never like to do that unless it's absolutely necessary, because there's so much into individual variation. I think it would look like a mess.
So otherwise, the input RNA you need for this experiment, especially if you use one of the available kits, is actually quite low.
Okay,
it's probably not as low as RNA-seq. I don't have any number in my head, but
yeah,
it's not like outrageous or massive. It's actually surprisingly low.
Oh, that's good. Because I know with like anything that has cross-linking, you tend to like lose some stuff along the way.
Oh, this is something else. Yes. So I basically showed 2 different kind of things. So the Riboseq experiment has no cross-linking in it. And this is the one where I mean there's the input RNA you need is relatively low.
Right.
But if you wanted to do the thing with the cross-linking, that's a different story. Because alone to set up the cross-linking conditions, it would, with these little gonads, I would feel sorry for the all the pogonas. Like, you know,
Yeah, fair enough. Thanks.
Hi, Susan. May I ask a question? It's Craig MacArthur.
Oh, hi. Yes.
Phil Dunker for a very good seminar. Many thanks. A really clear, really excellent seminar.
Danke fürs Zuhören. Thank you, Craig, for zooming
lovely
in.
Slides. You showed a slide about halfway through that had the 3 lines in it, the horizontal line, the vertical line. And you said with the horizontal line, if I got it correctly, the mRNA, the protein translation or whatever, would be controlled by mRNA levels. Is it possible, maybe this is a non-question, can it be controlled by controlling the ribosomal concentration, the ribosome concentration as well, or would that, the cell doesn't do that? Is that a
yes? I would
question?
say to control it on an mRNA level, I think it's probably more unusual, but it can happen. I think it can happen if you imagine that something happens to the cell.
Oh, that's a shame.
That for some reason the translation compensate on the mRNA level for it. So say the translation for some reason slows down by half. So you just produce 2 times more mRNA and you still have the same protein outcome. It's, but I think this isn't the more unusual case, to be honest.
Okay, okay, thank you.
Thank you.
Any more questions? I might have stunned them. Got a couple more minutes for questions. So what's the involvement of the tRNAs in this, and does their abundance influence the translation? Yeah. Can they be insured?
Oh, definitely.
Yeah.
Yeah, sure. They have a lot of influence, and also like codon, like one tRNA can decode more than one codon, and there's something like one codon, one amino acids can be made by different codons, and every codon has their, of course, tRNA. But yeah, anyway, so there's something like the optimal codon and the suboptimal codon, and then it also plays a role if like the sequence, the RNA sequence is made up of suboptimal codons or optimal codons, and there's a huge amount of regulation going on there as well. I think.
I'll just ask a question. Thanks, Arthur. So let's say someone's interested in something like a fish or an insect, or which is not a model, not a species which is well studied like Pagona, and we want to know it's subject to some stress, maybe a temperature stress, and then how that changes the regulation of proteins, whether looking at, so whether the messenger RNA is upregulated or downregulated, and then whether proteins are translated out from that. What's actually involved in a practical sense? I mean, you say it's easy, but I mean, how much time's needed, how much money's needed, et cetera, to do that, to do this sort of stuff in sort of round figures?
Yeah, so the first thing, of course, you would always think of RNA-seq first. And if you have already a set up RNA-seq experiment, you can very relatively straightforward go to the Riboseq as well. But if you don't have a set up RNA-seq, I think it's not too difficult to set it up, but you would need to know how much, I don't know if you talk of insects, how much insects you need to get enough RNA and stuff like that. But if you know all of that,
no, we don't know any. We don't know anything. So
you don't know anything.
I mean, is this? I'm trying to get a feel for this. Is something which is, you know, it's a major project to get to where you start doing this, or can you get insects? And as you know, insects are quite small. They weigh in the milligrams per individual. I
think it's rather straightforward. I would say it's rather straightforward and quick, because I think there must be already some literature out there. There must be already people who have tried RNA-seq on insects, because I have no idea of insects, so I would go there, read.
Oh, I'm sure. So you just need someone who's done RNA on any insect, not the species that you're actually working with.
For example, for me, because I don't work on insects, and I believe you have never worked in the lab with insects, it would be already the question how to get the RNA out of the insects. Is there something special needed because they have a lot of these structures or not? But I believe there will be protocols out there already. So if the protocols are out there already, then I would just go try to find the best one, and usually the protocols work reasonably well. So I would go from there, try to make a good RNA extraction, and once you know that, once you have this and you know how much RNA you can get, then to make an RNA-seq is almost no work, because there are kits nowadays out there, and even you can send your RNA to a facility and they do the rest for you. So the thing is really to get the RNA out of the insects. And to do a Riboseq experiment is equally easy. There's also kits, or at least there's one kit I know for it. The kits are always very easy to use, are straightforward. And this kit I know needs relatively low RNA input. So it's hard to give you a timeline, and I'm always estimating time totally. You have to make it ×5 when I say something, but but I would say this can be very quick. And from the time for the money, yeah, the sequencing. I think one lane nowadays costs $1,000 to $2,000, but I don't. But then you would probably need a higher amount of replication than you need for a cell line in the lab. Yeah, I couldn't estimate it would be not good if I estimate now the money. I wouldn't.
That's the cost of the library prep and everything as well.
Yes.
I'm just trying to think, the last quote that we got for like our gonad samples, do you remember what it was, Arthur? We had like quite a lot of samples, and it was like 80 odd samples, and we got a quote for like $15,000 thereabouts. And I saw someone in the comments asked like the amount of gonads that you would need, and in my case, most of the gonads that I'm sequencing from, I can't even see them. Like I literally go in blind. I know that there should be something in a tube because I put it in there when I dissected it, but I can't actually see it anymore. And I actually have really good success with that. And I have seen some of the eDNA people, I think it was Kat Campbell, I saw her grinding up insects in the lab at one point. I think the issue with them is because they've got exoskeleton and all this extra stuff that you don't want. There's maybe some extra steps in the protocols, but there are kits that would deal with like all the extra mess. And it's really not that technically challenging, the actual lab work component of it. Like once you get your insects, you've preserved them appropriately, put them through the protocol, you just send your RNA somewhere and then they take care of the rest. And it's really not that hard.
Yeah, your biggest challenge, Ben, would be that all of these processes are tissue specific, and in fact are even cell line specific. So you couldn't just mash up an insect and try and get an answer to the question that you're asking in relation to, thermal gradients through the environment. You'd have to target the different tissues at the very least, and of course that makes for very small samples. So then you probably just have to pool the samples across insects under each treatment, which isn't, that's not necessarily a problem. But you have to remember that the responses to temperature are going to be tissue specific in terms of the profile. So you'd need to separate the tissues out. All right. Well, I think we're at 12:30 now, and people have to move on. So allow me to thank Susan again for a really clear and enlightening talk, and really interesting to how much you can now delve into the cellular processes to answer questions that relate to environmental responses in species like Pagona, but also perhaps like some of the insect work that Ben's doing along environmental gradients. So it's really exciting times, great time to be a biologist, coming in as a biologist, I think. So thanks very much, Sarah, and look forward to catching up with you after the lockdown lifts.
Yes.
And everyone else.
Okay, thank you so much, everything, for zooming in and listening.